G proteins are important molecules in cell communication. When bonded to GTP, G proteins are in an active conformation that allows them to pass on signals. Once the GTP is hydrolyzed and a phosphate is lost, the GDP-bound G protein is inactivated. A G protein-coupled receptor has 7 alpha helices which pass through the plasma membrane. The binding site accepts signaling molecules from the extracellular space to activate G proteins inside the cell. A G protein has 3 subunits. The alpha complex is where GTP and GDP bind. The beta and gamma complexes are strongly associated with each other. Both the alpha and gamma complexes are tethered to the membrane by lipid tails. When GDP is bound, all 3 subunits stay closely associated. After a G protein has been activated by a GPCR and the GDP is replaced by GTP, the alpha complex dissociates from the beta-gamma complex, putting both into their active conformations. GPCRs can activate ion channels. In this example, acetylcholine activates a GPCR, which activates the G protein. The activated beta-gamma complex can then associate with the potassium channel, causing it to open. This allows potassium to pass through and pass on the signal. Secondary messengers are key components of cellular signaling. They relay, amplify, and spread intracellular signals. G protein-coupled receptors can trigger the synthesis of secondary messengers, such as cyclic AMP. Activation of the G protein-coupled receptor leads to activation of the alpha complex, which in turn activates adenylyl cyclase. This enzyme catalyzes the reaction of ATP into cAMP, which then carries on the signal by activating protein kinase A. pKa's nuclear localization signal is exposed, allowing it to enter the nucleus and promote transcription of certain genes, leading to changes in gene expression. G protein-coupled receptors can work with ion channels to pass on signals. In this example, the activation of a G protein-coupled receptor leads to the activation of phospholipase C through the alpha complex. Once phospholipase C is activated, it cleaves inositol phospholipids into inositol triphosphate and diacylglycerol. The inositol triphosphate, or IP3, then associates with a calcium ion channel in the ER membrane, allowing calcium ions to enter the cytosol. These ions then associate with protein kinase C. The diacylglycerol, which is tethered to the membrane, also associates with PKC. With both the diacylglycerol and calcium ions, the PKC is activated and can pass on the signal.